Hi, my name is Claudia. Today is February the 6th, 2020, and I'm doing week 4 lab for my microbiology class. This week lab is called Biochemical Testing for Microbial Identification. I'm going to be using the methylene red, Voget's Pashker, I can't say that correctly, and catalase testing for E. coli and also for staph or S. epididymitis. I think that's what it's called. So for that experiment, I have here, I have two MVP broth. Where are they? MRVP broth, and they're labeled. One is labeled S. epididymitis, and the other one is labeled E. coli. And I also have my cultures that was done in the nutrient broth right here. So I'm going to be performing my experiments with these. Okay, so the first thing I'm gonna do is gonna get my pipette out of the alcohol that I have here. I have to make sure I get all of it out, and I'm gonna be inoculating. It's hard to do this by yourself. Gonna be inoculating. I still can't see it. Maybe right there. Gonna be inoculating the lid. I'm going to get some of the broth from this active culture that I have here, and I'm going to be putting it into the MVPR or MRVP tube, and then I'm going to test tube, and then I'm going to let that grow for 48 hours. Also going to inoculate this one as well. These things, they smell really funny. I'm very curious to see what is gonna grow, because when I did my plates last time, actually nothing grew. So I was kind of really disappointed. So I hope this time I actually see some growth. Gonna be disposing of this in my little bleach container that I have over here. All right. Getting my other pipette out of the alcohol. And I'm going to basically just repeat the same thing here. All right, here we go. Now I'm gonna do this one. All right, so now I'm going to get there we go. Disposing of this, putting the lid back on, make sure it, oh well, I kind of messed that up. I was supposed to burn it. Well, let's do it just to do it. And then I'm gonna be doing this one as well. So on my MRVP, these two ones here, let me out that. I am going to be placing them into the incubation area, and I will have them there, and I will keep them there for 48 hours. Then I'm gonna come back and hopefully see some kind of growth. As far as these two go, they're gonna go back into the refrigerator. All right, let's see what else this lab's call for. So I have 4 test tubes. They're clean and they're labeled, basically 2 for E. coli and also 2 for staph. And after this 48 hours, I'm hoping to see some kind of growth. What I will do next is that I will separate these tubes, half of the E. coli in one and half in the other one. And I'm going to add to each one of those, I'm going to do the methyl red, which I have here. This is like a 2-part experiment. I'm going to add 7 drops to the first test tube of the staph and also the E. coli. And I'm going to see, I guess, observe the experiment as to see what happened when I put that in there. And on the second test tube, I have Barrett's A and Barrett's B. And I'm going to put 12 drops of A, I believe. I have to look at my thing. And then I'm also going to put part B into this other test tube here. And then I will observe the reaction on both. And I'll do that for both the E. coli and also for the staph. And I think that's it. Thank you.